This lecture will focus on the complex process of sperm production, sperm development, and sperm pathways. The male testicles produce the sperm cells. Let's dissect one of the testicles and see how the sperm production process occurs. Each testicle contains about 250 lobules, each having 2 to 4 seminiferous tubules. The sperm cells are produced inside of these seminiferous tubules. The seminiferous tubules are highly coiled and tube-like structures that converge to form a single straight tubule that leads to rete testis. This is inside the seminiferous tubules, where sperm cells are produced. The internal part of the seminiferous tubules comprises stratified epithelium, which consists of Sertoli cells and germ cells. Leydig cells are located just outside the seminiferous tubules and in between seminiferous tubules. Leydig cells are the primary source of testosterone, which regulates the sperm production. The entire sperm development process takes place inside the seminiferous tubules, and the process is called spermatogenesis. Let's take a closer look at how spermatogenesis happens. This cell right here is a diploid primary germ cell with 46 chromosomes. It goes through mitosis, producing 2 diploid daughter cells, each with 46 chromosomes. One of the daughter cells is a type A cell, which undergoes the self-renewal process, becoming a new germ cell to maintain the high productivity of the germ cells. The other daughter cell becomes a diploid spermatogonia and goes through meiosis 1, producing 2 haploid daughter cells with 23 chromosomes each. The 2 secondary spermatocytes undergo meiosis 2, giving rise to 4 spermatids, each becoming a sperm cell. Sertoli cells provide structural support and secret fluids to nourish the developing sperm cells, facilitate the movement of developing sperms, and protect the germ cells by forming tight junctions. These junctions form a blood-testis barrier to prevent substances in circulation from entering the inner part of the seminiferous tubules. Thus, the seminiferous tubules are in an immune-privileged environment, which means sperm cells are not exposed to the immune system. Under the influence of the FSH hormone, Sertoli cells produce androgen-binding proteins that keep testosterone in the lumen. Leydig cells are the primary source of testosterone, which is crucial for sperm development. When sperm development is complete, the sperms are moved through the lumen to the rete testis by peristalsis constriction by the smooth muscle around the seminiferous tubules. Now, millions of sperms are moved from these seminiferous tubules to the rete testis to the efferent ductals and finally to the head of the epididymis, where the sperm cells mature. During intercourse, mature sperms are pushed from the epidermis through the vas deferens to the prostate gland and into the urethra. In each ejaculation, up to 500 million sperm cells are deposited at the posterior vaginal fornix. And avoiding the female vaginal acidic environment, the sperm cells quickly attach to the cervix mucus and start entering the uterus. Only a tiny percentage of the 500 million sperm cells will survive and enter the uterus. Therefore, a man is considered to have a low sperm count if his sperm cells are less than 40 million sperm cells in each ejaculation. In this difficult journey, only about 5,000 sperm cells make it through the uterus, and about 1,000 sperm get a chance to enter the fallopian tube. Finally, only about 200 sperm cells reach the egg. Out of these 200 sperm cells that have made it to the egg, only one fertilizes the egg. As the sperm approaches the egg, they bind to the zona pellucida in a process known as sperm binding. This triggers the acrosome reaction, in which the acrosomal enzymes are released. These enzymes then start to digest the zona pellucida and allow the sperm to tunnel toward the plasma membrane of the egg. Finally, the haploid nuclei of the two gametes come together to form the genome of a new diploid zygote.